Hello everyone! Today I'll introduce our work for the Bio+MedVis challenge. Our goal is to analyze and visualize 3D microscopy data to study interactions between combinations of biomarkers. Our approach tackles three main tasks: detecting regions with specific biomarker combinations, showing these regions in the global tissue context, and enabling detailed exploration at the cellular level. Using a set-based approach, we select marker voxels, isovalue, tile by tile, to find co-expressed regions. We generate protein masks, find overlaps to reveal co-expressed markers, and run this on fine tiles for cellular detail. On the global view, multiple channels can be enabled simultaneously. Here we select MART1, CD31, CD8A, and CD103. Users can adjust color mappings and intensity ranges to emphasize different proteins. Users can zoom in and out across multiple resolution levels. Zooming in reveals higher resolution details for local regions. Users can rotate and explore the 3D image, while zooming out shows the full tissue. In the top left status bar, we display the current zoom level, resolution, and active channels. In this view, MART1 highlights tumor cells, CD31 shows blood vessels, CD8A marks cytotoxic T cells, and CD103 identifies tissue-resident effector T cells. This lets us see where the tumor is, how immune cells are infiltrating, and how blood vessels support both the tumor and immune response. To guide exploration, we add some analytical views. Here, we are looking at tumor and immune cell interactions. First, we focus on MART1, which highlights tumor presence. The filtered upset plot, which shows intersections between multiple marker sets, reveals co-localization with MHC1. The filter panel lets you choose which channels are included in the upset plot calculation. Next, we turn our attention to CD8A, representing cytotoxic T cells. Expanding the upset plot reveals that CD8A frequently appears with CD103, which may indicate tissue-resident memory T cells, rather than circulating ones. We also highlight CD103, using the search functionality to quickly locate it. And then CD31, which marks cells in blood vessels. With all four markers, we focus on regions where CDA8A and CD103 densely co-locate by deselecting the DIST checkbox for MART1 and CD31, which affects heat map computation. Here, a grayscale heat map provides an overview of co-expressing regions, guiding us toward the areas with the most prominent interactions without interfering with channel coloring. Then, we select a tile, where surface rendering reveals finer structural details. Lowering the opacity of MART1 allows us to see the rich co-localization of CD8A and CD103 within the tumor microenvironment. Switching to the volumetric view, we put this tile into context, observing how these cells are distributed in 3 dimensions. Finally, by clicking on a smaller tile, we drill down to the cellular level. This reveals intricate spatial relationships between tumor cells and immune populations. Here, some T cells appear to be tissue-resident, marked by CD103, white, rather than entering from the bloodstream. This is a preliminary observation, showing how marker selection, heat maps, and volumetric visualization can help explore the tumor microenvironment. To make the results easier to interpret, we integrate semantic explanations powered by Biomni. Biomni is an AI agent for biomedical reasoning. When you click the button, it takes the current visualization and marker context and returns a short summary: what is visible, its biological meaning, possible regions of interest, and even potential clinical relevance. The current view highlights tumor cells, blood vessels, and distinct immune populations, including cytotoxic and tissue-resident T cells. Their spatial relationships, such as CD8A and CD103 co-localizing near MART1, suggest active immune tumor interactions that may be clinically relevant for immunotherapy. Our system helps detect where biomarkers interact in large 3D datasets. We combine volume and surface rendering with tools like upset plots and heat maps to explore the data. By tiling the volumes, we can spot co-expressed proteins from overview down to cellular detail. Right now, spatial binning limits detail, but in the future we'll use cell segmentation and deep learning for more precise analysis and interactive search. Thank you.